Okay, this should be the last recorded lecture for Chapter 2. This is slide set 2B, and we are covering lipids now. The difference between a saturated fatty acid and an unsaturated. In a fatty acid, you have this long chain of carbons, and in between each carbon atom is a bond. And if all of the carbons are bonded by single covalent bonds, that means every carbon atom shares only a single pair of electrons with another carbon atom. So if there are only single covalent bonds between the carbon atoms, that's going to be a saturated fat. Some examples of saturated fats are animal fats, butter, lard, like bacon grease. These are saturated fatty acids. They tend to be solid at room temperature. And you can see that it doesn't show you. This would be a double bond here between a carbon and an oxygen, but I still don't like the way that looks. Let's go back and see. Here we go. All right. So what we're looking at in this triglyceride, just focus on the carbons in that fatty acid chain. You see how every bond is just a single bond. That means only one pair of electrons is shared between each of those carbon atoms. So that is a single bond, single bond. If any of those bonds were double bonds between those carbon atoms in that fatty acid chain, then this would be an unsaturated fatty acid. So this is saturated. There are no double bonds. And then unsaturated means there's at least one double bond. So here we have one double bond, which causes the fatty acid chain to be bent. And that will cause the unsaturated fat to be an oil at room temperature, like olive oil. Okay, so only one double bond, and it's going to be an unsaturated fat, which are healthier for us. A trans fat is a modified unsaturated fatty acid that has been modified to take out the double bonds and make it a fat that ordinarily would be a liquid at room temperature. Now it is a solid. Trans fats are considered particularly unhealthy, even more unhealthy than saturated fatty acids. So trans fats can be found in things like margarine, peanut butter, not the natural peanut butter that you find in the grocery store that most people don't seem to like. Natural peanut butter has the unsaturated peanut oil in it, and you have to stir it to mix the oil in with the peanut part to make a paste. But if you get regular peanut butter, just regular peanut butter is made with it converts that peanut oil into a trans fat. And so the texture of it is different. It doesn't have a layer of oil that sits on the top. The fat in the peanut butter remains solid at room temperature. And most people tend to like peanut butter that's made with the trans fat. So that is not something that we found. A lot of people haven't made the change, to healthier peanut butter, because it just seems kind of, people kind of find it unappetizing to see that layer of oil on the top. And you really can't eat it and the natural peanut butter unless you stir the oil into the peanut part. If you just pour the oil off, it's just too thick and you can't spread it. So anyway, those are some examples of trans fats. Sometimes I tell my students about, because I'm older, 2 years from now, and I'll be able to retire. I don't know if I will, but I will be able to. So that lets you know that I'm not young anymore, but I'm not old either. But anyway, when I was like 5 years old, I lived in the time before McDonald's changed the fat that they use to fry their French fries in. Now they use an unsaturated fat, like a vegetable oil, I think it's canola oil. I looked it up one time, but when they used trans fats to fry their French fries, and a lot of people really love McDonald's fries, but you have never lived until you had French fries back when McDonald's, when they were less healthy. I'm not saying McDonald's is healthy. I'm just saying that they used to use trans fats, and then trans fats were outlawed in fast food restaurants. So I remember the French fries. I remember when my brothers and I were little kids, it was a treat just to go to McDonald's and get McDonald's fries. I mean, we were happy just to get just an order of small fries because they were so good. I can still taste them. So, but they're not healthy. So McDonald's went a few years later and changed from using trans fats to fry their French fries in to using the healthier, still probably not the healthiest, but using the healthier unsaturated fat, the canola oil, vegetable oil mix that they use now. And their fries are still good, but they're just not like they're not as good as what they were. And omega-3 fatty acids are heart healthy. So you can look up foods that contain omega-3 fatty acids. I think like salmon is one of them. And I think avocados, actually. I know avocados are considered like one of the heart healthiest foods you can eat. So I'm eating half of one today for lunch. They have high calories though. But anyway, moving on, we discussed triglycerides and we discussed how they can be saturated or unsaturated. And then now we're going to move on to phospholipids, which are similar to triglycerides, but there's a few differences. So instead of having 3 fatty acid tails, they only have 2. They've got 2 fatty acid tails. And in addition to the glycerol that's attached to the fatty acid tails, there's also a phosphate group. And this is what they look like. This is what we find in our plasma membranes of our cells. We have the phosphate group here, which is purple. You can see phosphorus attached to 4 oxygen atoms. But this is our phosphate group, and then our glycerol under that. And our phosphate group area is attached to the glycerol. That is called the polar head. And the polar head is hydrophilic or water-loving or water-soluble. So the heads of the phospholipids are either facing the outside of the cell or they're facing the inside where they're in contact with fluid. The tails face each other. So there's 2 layers of phospholipids, and the tails of each layer face each other, turn inward and face each other, because they are fatty acids, which are lipids, and they are nonpolar and insoluble in water. So this is what the plasma membrane is going to be like, is a double layer of phospholipids. We call it a phospholipid bilayer, where the fatty acid tails face each other because they are nonpolar and hydrophobic, meaning they're insoluble in water. And then the polar phosphate heads are hydrophilic and they face away from each other. Steroids are also lipids, and we mostly think of steroids as our steroid hormones. But all steroid hormones start out as a cholesterol molecule. Then the cholesterol molecule is modified. So cholesterol is important. We don't need too much cholesterol, but we need some cholesterol in our diets because we need cholesterol in our plasma membranes also, in addition to phospholipids. The cholesterol is the starting material for the synthesis of vitamin D and steroid hormones, as well as bile salts. And the steroid structure looks like 4 rings. 3 of them are hexagons and 1's a pentagon, but just remember the 4-ring structure. So all steroids have that 4-ring structure. Now, eicosanoids are derived from fatty acids, and the most important ones are prostaglandins. Prostaglandins play a role in blood clotting, controlling blood pressure, inflammation, and labor contractions. but they also will increase inflammation. So the inflammatory action of prostaglandins is blocked by NSAIDs, which are non-steroidal anti-inflammatory drugs like ibuprofen and aspirin. So NSAIDs block the inflammation caused by the release of prostaglandins. But those are probably the most well-known eicosanoids is prostaglandins. All right, then we've covered of our organic molecules, our big groups of organic molecules, we have covered carbohydrates and lipids, and now we're going to talk about proteins. Proteins are very large, and we are composed of a large amount of proteins. Proteins comprise 20 to 30% of our cell mass. They also have a large variety of different functions. We have proteins for structure. We have enzymes and hormones that are made of proteins. The hormones that are not steroids are usually made of proteins or amino acids. And then we have proteins that function in contraction in our muscles. Proteins also contain carbon, hydrogen, and oxygen like lipids, but proteins also contain a lot of nitrogen and sometimes sulfur. And sometimes they can even have phosphorus, but phosphorus is really more strongly associated with DNA than proteins. Now, the bonds between amino acids, because remember, amino acids are the monomers of proteins, they're the building blocks. So every time an amino acid is linked to another amino acid, that bond is called a peptide bond. Now, when proteins form, the first thing they do is the amino acids join, and then after you have this chain called a polypeptide, that chain of amino acids undergoes many structural changes, and so it has a very complex structure and shape, which we'll talk about in a minute. Now, there are 6 different types of proteins that we find. There are structural proteins. There are enzymatic proteins that speed up chemical reactions. There are transport proteins. For example, hemoglobin in our red blood cells transports oxygen. There are contractile proteins. The two mostly that we learn about are myosin and actin. Myosin has to attach to actin and pull on it, and that will cause muscle contraction. Then there's communication proteins. Mostly those are going to be hormones like insulin. And then there are defensive proteins like antibodies that are produced by our certain lymphocytes or white blood cells that will help us to have immunity against certain diseases. There's certain pathogens. All right, there are 20 different types of amino acids. Each amino acid is joined. You have a long chain of amino acids that forms a protein. The individual amino acid looks like this. It has a central carbon, and then it has an amine group, which is nitrogen bonded with 2 hydrogen atoms. Then there's just a hydrogen group bonded to that central carbon, and then an acid group, which is COOH. That's our carboxylic acid group or our acid group. And that's where you get the term amino acid, is from the amine group and the acid group. Now, what makes each of the 20 amino acids different is they each have a different group here. We call it an R group, but it will be something different for each of the 20 different amino acids. So when the 2 amino acids are joined by dehydration synthesis, the bond formed is a peptide bond. Now, the structural level of proteins is primary, secondary, tertiary, and quaternary. The primary structure is simply the sequence of amino acids. The secondary is that chain of amino acids will either spiral and form an alpha helix, like DNA, or it will pleat, like if you ever took a sheet of paper and formed a pleated fan, those are pleats. So sometimes the amino acid chain will pleat and form a beta pleated sheet. The alpha helix or beta sheet is the secondary structure. Then you take the secondary structure and form it into a three-dimensional structure, and that is the tertiary structure. That's the overall three-dimensional shape of the protein. And quaternary, only some proteins have a quaternary structure, but that is when you have more than one tertiary structure that is chemically combined. So this is showing you the levels of structure. We have the chain of amino acids, that's our primary structure. Then we have either the alpha helix or the beta pleated sheet is our secondary structure. Then we have the overall three-dimensional shape of the protein is our tertiary structure. And then two different tertiary structures, two or more, chemically combined is the quaternary structure. The animation that explains protein structure, the 4 levels of protein structure, is found in slide 54. Now, there's also a section that talks about how proteins denature. Proteins can denature if they are exposed to high heat or if the pH changes. Different conditions can denature the protein to where it won't function anymore. Denature just means it changes shape. So those 4 levels of structure are extremely important. Some proteins are enzymes, which speed up chemical reactions. They're biological catalysts. And the shape of the enzyme determines whether or not it will carry out its chemical reaction. So that shape is very, very important. The way enzymes work is they lower the activation energy required for a chemical reaction. So by lowering the energy required for that reaction, they cause the reaction to speed up. So in that way, they're catalysts. This is an example of an enzyme. For example, this is an enzyme that combines amino acids. So the enzyme is the purple molecule. The 2 amino acids fit into a shape on the enzyme, and then they are chemically combined and then released, and the enzyme can be reused. The enzyme is unchanged. It can be reused to join 2 more amino acids. Okay, our last section is on nucleic acids, and the monomers of nucleic acids are nucleotides. 2 different types of nucleic acids are DNA and RNA. DNA contains the nucleotides adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, which means they have 2 rings. And cytosine and thymine are pyrimidines, which means they have a single ring. There are 2 strands in DNA, and always adenines on one strand will pair with thymines on another strand. So A's will pair with T's. And cytosine, or C, on one strand will pair with guanine, or G, on the other strand. So that is complementary base pairing. A always pairs with T, G always pairs with C. So that's how you know, if you know the code on one strand, you can tell me the code on its complementary strand. So DNA, it has 2 strands that are joined together like a ladder. The rungs of the ladder are the bases, and then the sides of the ladder are phosphates and sugars. So each nucleotide has a phosphate and a sugar combined with one of those 4 bases, A, C, T, and G. And so the sides of the ladder are the phosphates and the sugars, and then the rungs or steps of the ladder are the bases. And then the ladder is twisted into a double helix. Now, RNA is also used to kind of assist DNA in making protein, but the RNA is going to be a little bit different. It's going to be single-stranded. The sugar is different. It's not deoxyribose, it's ribose. And instead of a T or thymine, RNA contains U or uracil. And here's an animation on DNA and RNA. I encourage you to watch it because I kind of rushed through this. This is on slide 68. And then I also encourage you to read about ATP, but I'm going to end here. This section on ATP is just a few slides long, but you do need to know about ATP, which is the molecule that we use for energy, and it's called adenosine triphosphate.